This video will provide an overview of the steps used to extract a plated culture of adherent cells from metabolomics. The video describes steps after the sample has already been aspirated, quenched, and frozen. For instruction in performing those steps, viewers should consult with the staff of the Michigan Regional Comprehensive Metabolomics Resource Corps. Step 1. Assemble supplies. All needed supplies should be assembled before beginning cell extraction. These items include dry ice to keep plates of cells frozen before extraction, wet ice to keep cells and extraction solvent cool, a cell scraper, which is a plastic device used to physically disrupt adherent cells and remove them from the plate, a benchtop microcentrifuge, preferably refrigerated to 4 degrees C, for pelleting and cell debris, pipettes, microcentrifuge vials, and autosampler vials to contain and manipulate the samples, and finally, the extraction solvent, which is used to lyse the cells, dissolve metabolites, and precipitate proteins and other macromolecules. Typical extraction solvents for polar metabolites are organic solvents such as methanol or acetonitrile, or a mixture of these solvents with water. Extraction of nonpolar metabolites such as lipids requires nonpolar solvents such as chloroform or methyl tertiary butyl ether. Today, we are using an extraction solvent composed of 8 parts methanol, 1 part chloroform, and 1 part water, which provides a balanced extraction of polar and moderately nonpolar metabolites. Depending on the analysis, internal standard compounds may need to be added. Consult with Corps staff for specific recommendations regarding preparation of a suitable extraction solvent. Step 2. Add extraction solvent. To begin the extraction, remove the frozen cell plate and place it on the surface of the crushed ice. Immediately add extraction solvent. For the 10 centimeter plate shown here, 1.5 milliliters of extraction solvent is used. For a 6 centimeter plate, 500 microliters is typical. Increase or decrease the extraction solvent volume in proportion to the surface of the plate. Step 3. Scrape cells. Scrape the entire plate several times with a cell scraper to release all cells from the surface. Once all cells have detached, tilt the plate slightly to one side to pool the extraction solvent along the edge. Use the cell scraper to move as much residual solvent as possible. Step 4. Collect solvent. Using a pipette, collect as much of the extraction solvent as possible and transfer it to a microcentrifuge tube. The solvent will contain some cell debris, and some cell debris will also remain in the plate. The residual cell debris can now be collected by rinsing the plate with additional aliquots of extraction solvent, but it isn't necessary for metabolomic studies. Store this tube in ice. The plate can now be discarded. Step 5. Centrifuge. Once all cell plates have been extracted, centrifuge the microcentrifuge tubes at 15,000 times gravity for 5 minutes to pellet all cell debris. Step 6. Remove supernatant. After centrifugation is complete, remove the vials from the centrifuge. Collect a measured volume of supernatant using a pipette. The supernatant can be transferred directly to vials for analysis or dried down using a vacuum centrifuge or a gentle stream of nitrogen gas. Whether the samples are dried or not, they should be stored at -20 C or lower if analysis will not be performed immediately, and they should be shipped on dry ice. For samples being sent to the Metabolomics Corps for analysis, please contact Corps staff for specific instructions about extracting, drying, or shipping your samples.